In this movie, we consider the molecular structure of potassium channels. Potassium channels play a fundamental role in shaping the electrical features of neurons. They are responsible for generating the resting potential of cells. They also act to shape the action potential, as we have seen from voltage clamp studies of the squid giant axon. They operate in very subtle ways to regulate the overall excitability of neurons, features that we have not yet discussed. The point is that to achieve the large variety of roles they play, there are hundreds of different kinds of potassium channels. In this movie, we first consider the basic molecular structure of potassium channels and compare them to the structure of sodium channels. We then discuss how and why so many different types of potassium channels can be created. Finally, we consider the structure of the selectivity filter, which enables the channels to be selectively permeable to potassium. The first potassium channel that was cloned was the Shaker potassium channel in the fruit fly, Drosophila melanogaster. When the sequence of the Shaker potassium channel was determined, it was found that the protein is encoded by one gene and was called the alpha subunit. The alpha subunit is in some ways strikingly similar to the sodium channel, and in other ways is strikingly different. The features that are similar are that the alpha subunit of the potassium channel has 6 membrane-spanning regions, S1 to S6, with one S4 region that is the voltage sensor, and a pore formed by the loop between S5 and S6. The alpha subunit of the potassium channel is basically the same as one domain of a sodium channel. Recall that the sodium channel has 4 domains. Each domain has 6 transmembrane segments, S1 to S6, with an S4 segment that is the voltage sensor, and a pore formed by the loop between S5 and S6. Also recall that the entire sodium channel is produced by a single gene. In contrast, genes that code for the potassium channels only transcribe messenger RNA that produces one alpha subunit. An individual alpha subunit cannot by itself form a channel. Rather, the Shaker potassium channel is formed by combining 4 alpha subunits into a tetramer, as shown here. There's one subunit, there's another subunit, there's a third subunit, and there's a fourth subunit. Indeed, it is now known that all potassium channels are formed from 4 subunits, where each subunit is encoded by one gene. The sodium channel is similar, except that the single polypeptide is folded such that each of the 4 domains is similar to the subunits in the potassium channel. 1 domain, 2, 3, and 4. In short, the potassium channel, like the sodium channel, has 4 subunits or domains, where each subunit is composed of 6 transmembrane segments, S1 to S6, where the S4 segment is the voltage sensor, as in sodium channels. However, unlike sodium channels, where the entire channel is encoded by one gene, only one domain, that is a subunit, of the potassium channel is produced by a single gene. Potassium channels in the brain show enormous functional diversity. Hundreds of different potassium channels have been discovered. Some has fast activation, whereas others are slow. Some are activated even at highly depolarized membrane potentials, whereas others require greater depolarization to be activated. Some even have inactivation gates, just like sodium channels, whereas others do not inactivate. Some of the channel diversity is a consequence of the different genes that encode different potassium channels. Mammals, for example, have 17 voltage-gated potassium channel genes. Indeed, the potassium channels in the squid giant axon, for example, are produced by a different gene than the homologue of the Shaker potassium channel. But there is another factor that accounts for potassium channel diversity: alternative splicing of the RNAs transcribed by the potassium channel genes. Recall how alternative splicing works. The DNA of all genes contain expressed sequences called exons, which code for the protein, and non-coding sequences called introns. Because RNA is synthesized from this DNA, the RNA, called pre-mRNA, also contains the exons as well as the non-coding introns. During the production of the mature mRNA that encodes the protein, the introns are cut out and the exons are spliced together to produce a competent mRNA that is translated in the cytoplasm. The splicing can produce different combinations of exons and different mRNAs. The top splice variant is the same as was shown in the previous slide. All 4 exons comprise the mature mRNA, which produces a particular polypeptide. 2 different splice variants are shown in the lower portion of the figure. In one, labeled splice variant 1, only exons 1, 3, and 4 are incorporated into the mRNA, while the exons 1, 2, and 4 are incorporated into splice variant 2. Consequently, different proteins will be expressed by each splice variant. The idea is that a potassium channel gene, the gene for Shaker is just one example, can produce several different alpha subunits through alternative splicing. The different subunits produced by the alternative splicing of a gene is a major factor for generating the functional diversity among potassium channels. To illustrate this diversity, consider that a potassium channel can be constructed from 4 of the same alpha subunit types produced by one gene. Such a channel is called a homomeric channel. Of course, a variety of homomeric channels can be constructed from subunits produced by different genes, as illustrated by the 2 channels composed entirely of white or entirely of black subunits shown in the top part of this figure. But a potassium channel can also be formed by combining 2 or more different types of alpha subunits, subunits created by alternative splicing of the RNA produced by one gene. Channels composed of different subunit types are called heteromeric channels. 2 heteromeric channels are shown in the lower part of the slide. The one on the left is composed of 3 splice variants, each indicated by a different color. The one on the right is composed of only 2 splice variants. You can imagine the large number of different potassium channels that could be produced by combining different splice variants generated by a single gene. Each combination of subunit types would produce a potassium channel with unique functional properties. Next, we turn to the Pour loop and the selectivity filter that endows the channel with its ability to selectively conduct potassium and not sodium or any other ion. The operation of the Pour and how the selectivity filter in the Pour endows the channel with its ability to pass only one type of ion, potassium in this case, was shown by MacKinnon and his colleagues. This was a stunning achievement, and MacKinnon was awarded the Nobel Prize in Chemistry in 2003. MacKinnon and his colleagues used a bacterial potassium channel called KCSA. The exact structure of the channel is not terribly important for our purposes, but I would like to describe it just so you understand how it is put together. The only important part for us is the selectivity filter. This channel has a simple topology and is composed of 4 subunits, each one indicated by a different color in the slide. Like other channels, the subunits surround a central pore. Each subunit in turn has only 2 segments, an outer helix or segment that faces the lipid membrane and an inner helix that faces the central pore. The outer helices of 2 subunits, the green and yellow subunits, are indicated by the arrows, as are the inner helices of the blue and red subunits. The 2 segments correspond to the S5 and S6 segments of the Shaker potassium channel. This particular bacterial potassium channel does not have an S1 through S4, so it has no voltage sensor because it is gated by acidity rather than voltage. This slide shows a drawing of the channel as viewed from the side. The gate is on the bottom of the channel. The channel has a water-filled central cavity. The outer and inner helices or segments are shown. Of particular importance is that the inner and outer segments or helices are separated by a series of amino acids that form the pore helix and the selectivity filter. The pore is shown as red lines here near the top of the channel, and the selectivity filter that lines the pore is shown in yellow. I would like to emphasize a most important feature of the selectivity filter. The sequence of amino acids in the selectivity filter is the same in the pores of all potassium channels. The entire sequence is composed of 4 pore loops inside the pore, 1 loop from each of the 4 subunits. MacKinnon called these amino acids the potassium signature sequence, and was so profoundly struck by this universal feature of potassium channels that in his 2003 Nobel address he wrote, "The conservation of the signature sequence amino acids in potassium channels throughout the tree of life, from bacteria to higher eukaryotic cells, implies that nature had settled upon a very special solution to achieve rapid, selective potassium conduction across cell membranes." So let's explore the selectivity filter in a little more depth to see how it works. A more detailed view of the filter is shown in panel B1 in the lower left of the figure. The 2 helices are shown, as is the pore helix. Notice there are 4 potassium ions in the selectivity filter. Panel B2 shows a skeletal version of the filter. The point here is that there are 4 binding sites, each binding 1 potassium ion. This is a side view of the filter. Next, let's look down upon the binding sites, the topmost site, which is shown in panel B3, just like the view Mr. Magoo is seeing. Here you can see 1 potassium ion bound in the binding site that is formed by the 4 subunits. The way the potassium ion is bound in the binding site is shown in the lower portion of panel B4. This requires some additional explanation. When ions are in solution, they're hydrated and surrounded by water molecules, as shown in the top portion of B4. The hydrated potassium ion is too large to fit into a binding site of the selectivity filter. Before the potassium ion can enter the filter, it has first to be stripped of its water covering. That, as we shall see in a moment, is the job of the pore helix. Once the water is stripped from the potassium ion, the size of the ion fits perfectly into the binding site of the filter and is stabilized by the negatively charged carbonyl groups of the filter. To see how this all spins together, I will use a drawing of the channel shown in the top right. This is a cross-section of the channel that shows only 2 of the 4 subunits. The outer helix and inner helix are shown, as is the selectivity loop and the selectivity filter. The pore helix is part of the pore loop that is connected to both the inner and outer helices, similar to the way the pore loop connects segments S5 and S6 in other potassium channels and sodium channels. The point to be noted here is that the portion of the loop pointing into the vestibule is negatively charged. When the hydrated potassium ions move into the vestibule, the negatively charged pore helix strips the water molecules from the hydrated potassium ion, allowing it to bind to a binding site in the selectivity filter. So let's put it all together and see how the selectivity filter works. The gate is closed, so intracellular potassium cannot enter the vestibule. There are 4 potassium ions bound in the selectivity filter. As soon as the gate opens, however, the concentration force drives potassium into the vestibule. The negative charges on the pore helix then strips the water from the ion. The electrostatic force on the stripped ion then drives the potassium from the lowermost binding site to the next site. The electrostatic force exerted by that potassium ion then drives the ion in the next binding site upward, and so on until the ion in the highest binding site is driven into the extracellular fluid. So long as the gate is open, hydrated potassium ions are continuously driven into the vestibule, stripped of their water, and the process is repeated. It's a bucket brigade. In short, there is an outward flow of potassium ions from the inside to the outside of the cell, driven by the potassium concentration difference, where the selective flow of potassium ions is due to the operation of the selectivity filter.